For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button because we are just getting started. Okay, looks like we are live tonight, late night on Standing for Truth. We got another good one. This is definitely going to be a must watch. As always, we have a lot of fun with these evolutionists. We have a lot of fun with the critics. As I always say, they give me job security. I could probably do this for another million years. So tonight we are educating Joshua Swamidass on genetic diversity. Also, we're going to play a clip from one of our favorite critics, Dr. Dan the Pseudoscience Man. Some of you may know him better as Damage Control Dan. So we'll play a clip by him, and we are going to show conclusively that the junk DNA paradigm has been overturned. So right here, we've got our beautiful thumbnail, SFT versus Swamidass. So this is going to be a lot of fun. I've got his arguments pulled up. We'll screen share that, of course, and go through it bit by bit. You guys will probably notice that oftentimes with these critics, with those that believe so desperately that all life has descended ultimately from a single-celled organism billions and billions of years ago, ladies and gentlemen, they proudly proclaim that whales and banana plants are related through common ancestry, and they think that this is science. Okay, they promote this as empirical science, so can't make this stuff up, guys. It's going to be a lot of fun. Anyways, before we get right into it, Thursday, 3 p.m. EST, interview with Dr. Jonathan Sarfati, one of my favorites. I've been listening to him for years. He is a warrior. He's a beast. When you listen to him, he's definitely a genius. So we're going to be touching on evidence for creation, refuting the critics. And if you've seen our past interviews, for example, with Dr. Rob Carter, Dr. Rob Stadler, who was with us to debunk Professor Dave. There it is right there. Definitely check that one out. There's the Dr. Carter one. Dr. Jackson, of course. All great interviews. But what we've made certain to do is that we touch on a number of different topics. So with Dr. Carter, we touched on Adam and Eve, genetic entropy. When Dr. Rob Stadler was on, we demolished good old Professor, quote unquote, Dave on abiogenesis and Dave's attack on Dr. James Tour. When Dr. Jackson was here, we touched on a number of topics, including junk DNA, chromosome 2 fusion. So for Thursday, make sure you're here in the audience. It's going to be very interactive. We'll have a Q&A for Dr. Sarfati. We're going to touch on evidence for a young world. We're going to go over a number of lines of reasons why abiogenesis is not even remotely plausible. So we're going to have some fun. We're going to have some fun, guys. So I want to thank everybody who's supporting this channel, all of our donors, everybody who supports us with super chats. You guys are awesome. Can't thank you enough. It's because of your amazing support at the end of the day why we are able to be so active on this channel, putting out daily videos, streams, discussions, interviews, debates. So we've got a lot of good interviews in store for you guys. We are working overtime to get the biggest names in young earth creation on this channel to refute the critics. So good to see everybody in the chat. Doki Doki, thank you so much for the super sticker. Lena Powell, good to see you as well. Doki Doki says, Heart, chess master Sarfati. Yep, I've heard he's a world champion, and he's even defeated numerous at the same time blindfolded. I mean, he would destroy me in chess. I am not good at chess at all. So yeah, definitely be there for that, guys. And why don't we just get right into tonight? It's going to be interactive. We got some videos. We've got some videos we are going to cover, some articles. Yeah, it's gonna be a lot of fun. So why don't we get right into it? Let's show you. So we've got Joshua Swamidass here. Now, apparently these were some objections or questions he had following the must-watch Dr. Carter interview. where he states, what is also interesting here is that, and I'll read through this and then we're going to spend some time on it, okay? So it's going to be a lot of fun. 1, humans are far less genetically diverse than other species, Swamidass says. Carter correctly notes this. Yes, we've been saying this for years. This was a direct expectation from the biblical creation model. Think about it. If we've descended from just 2 people, Adam and Eve, thousands of years ago, okay? And I've been asking this question. I've been putting forth this empirical data for the 2 years that I've done this, the 80 debates roughly that I've had, informal and formal. But if we've got 2 people, Adam and Eve, that we've descended from ultimately, what are we going to expect? Well, naturally, we would expect low genetic diversity in human beings as compared to a higher level of genetic diversity, okay? But for Swamidass, who holds to fish to fisherman, pond scum to people evolution, if that type of evolution were true, and we've been accumulating, okay, we've been accruing all these genetic mistakes, mutations, typographical errors in a text for millions and millions of years, obviously it doesn't take a genius to conclude that what we would expect is high levels of genetic diversity. Evolutionists didn't predict the low levels of genetic diversity in humans. There was every reason, based on what we know about the story of evolution, that we should have expected high genetic diversity. And yet the data that we have confirms biblical creation expectations. Okay? How do evolutionists explain the low genetic diversity in human beings? Well, we know that they've resorted to a post hoc, ad hoc explanation, which we will get into later. But that's just a brief explanation for those that have not already heard me explain this a million times. It can be redundant, but it's important for the critics, because oftentimes it's in one ear and out the other. And here's the thing, though. We can easily do 2, 3, 4-hour videos on these critics, because you'll notice that in one paragraph, okay, they'll say more things wrong, or they will use more straw man arguments or use more misrepresentations in that one paragraph than there are words in that paragraph. So it oftentimes takes a lot of work refuting every single claim and every single assertion that they make. Okay, so his second point, he says, due to the flood bottleneck, young Earth creationists should predict that humans are more diverse than any group of land species. More diverse, he says. In brackets, he says 10 human genomes on the ark versus 4 genomes per kind. He then says Carter doesn't really note number 2. So obviously he's not up to date on the literature. This video, I was debating on titling it 100% proof that the critics don't read creationist literature, but I think it speaks for themselves. So he says, so why is it, Swamidass says, that there is less diversity in humans than, for example, the horse kind, or maybe the ape kind, the rodent kind, the felid kind? I don't think there is any good explanation for this, Swamidass says. You can't make this stuff up, guys. And it is particularly interesting that Carter doesn't address this. It's particularly interesting that Swamidass clearly has not read very much of young Earth creation literature. I doubt he's up to date on the latest books coming out of the young Earth creation, young Earth creation scientists, for example, like Contessa Bones replacing Darwin, Evolution, Achilles' Heels is very good as well. So we're really gonna touch on this one, guys. So that was just a brief overview of what he said, and we're gonna get right into it. Doki Doki says, no refutation, so let's build a straw man with Dr. Swamidass. Yeah, instead of build your own hominid, Doki Doki, build your own straw man. I like it. Okay, so if you guys didn't gather just by reading that, this is going to be an easy one. Okay, so let's get right into this. Okay, we touched on it briefly. What should we expect given the starting point of biblical origins and the design diversity hypothesis? Okay, here's the thing, and I point this out in every single debate. And it's funny because even when you debate the same people, for example, David Neff comes to mind, or Adam Heap, he's a zoologist, herpetologist. I've done numerous debates with these guys, as well as others. But you'll find that they'll use an argument in, say, debate number one, and then you correct them and say, no, we explain the origin of genetic diversity as the result of divine creation, created heterozygosity. R.J. Downer's favorite phrase, of course. And then you'll debate them again, hoping that it's a profitable discussion, one that's just not going to be talking past each other. And then they use another straw man argument. And then you ask them, is this, I think it was this, actually, it might have been the third debate, third or fourth with David Neff, where I said, how do we explain the origin of genetic diversity versus how you do? And it appeared that even after 3 debates and 4 debates worth of explanations from myself, they had a hard time reiterating it. And, it's not just David Neff's fault. This is a common occurrence. So that's why the most important question to ask is, where does genetic diversity come from? Tony Lincoln, good to see you. Good to see you. Thanks for being here. I appreciate it. Hello to you as well. Tony Lincoln says, they don't listen, do they? No, it's in one ear and out the other. Because remember, they're not questioning their basic presuppositions. They're not questioning their worldview. Okay, so they're here to push their science fiction religion of pond scum to people evolution on us. They're not expecting arguments from our side. And then all of a sudden they come into a debate unprepared, and then they are forced to refute the overwhelming evidence for our position. So they have to pretend like it's not there. They have to pretend that we did not provide these arguments. So that's why it's in one ear and out the other. So good point. Good point, Tony. So the most important question to ask then is, where does genetic diversity come from? And this is a question I ask in nearly every debate. Because as I said, this way we can have a good debate where we are not talking past each other. Who wants to talk past each other the whole time? Dr. Carter actually makes a good point in our discussion on this issue. Okay, George Bond, good to see you, brother. He says, I'm walking the dog listening. Awesome. Awesome. I love when you're in the chat. You're a blessing. You've been an incredible help. George helped us get Dr. Sarfati for Thursday, so it's going to be a lot of fun. Tell your dog I said hello. Okay, so let's see. I'm going to pull up this video where Dr. Carter makes a good point in our discussion, actually, from a week ago on this issue, that what? Evolutionists typically argue against the creation model by what? What do? They assume evolution. They have essentially chosen to argue against a straw man. They argue against evolutionary creation and not biblical creation. We as creationists don't assume the vast majority of DNA differences are the result of mutations over time like they do. Okay. Before I go any further, let's show our first clip here. Brother Sal, good to see you. Good to see you. He says, greetings, SFT. This topic is, Lena says, hi, Sal. Sal, people are excited when you come into the chat. We love your work. You're an incredible blessing. He says this, so I'll learn from you today. God bless. God bless you too. It should be a lot of fun. Okay, guys, if you haven't seen Brother Sal's lecture we had last week on fitness, the worthless definition of evolutionary fitness, please check it out. It was rock solid. Okay, so let's share screen here, share audio. Boom. So you're going to see I've got a lot of links up here. So before I get started, I will say that Swamidass, he does say that he assumes created heterozygosity. Although he says he does, you can quickly tell based on his argumentations from the TMR4A, okay, time to the most recent 4 alleles, as well as the way that he uses arguments that doesn't assume that God would have placed these design variants in functional and useful and beneficial linkage groups. You quickly realize that he's just pretending. It's pretend with Swamidass. He wants to pretend to assume our position of created heterozygosity, but then proceed to use a straw man argument against that which he pretends to assume. Okay, so yeah, let's see this clip. You guys will see what I mean. I like how Dr. Carter is going to reiterate what I said here, but it really stuck out at me what he says. So
that a literal and Adam and Eve could not possibly exist. Basically, it comes down to genetic diversity. And some of the critics, including Dr. Francis Collins and Dr. Venema, claim that it would be unfeasible even and unscientific for a single human couple to have, existed ever together. And they would even say that it would require mutations so much are much too fast in order to account for all of the variety that we see in the human population today. Is there any validity to this argument?
Venema has said that, and Francis Collins has said that. In my article, Adam and Eve, I always get confused because the title isn't the same as the URL on I think it's called Adam and Eve Biologos on creation.com with the URL, but it's no, not that one. You showed it earlier. Yes, that's it. I pointed out here that they assume that all variation is due to mutation. So they assume evolution to critique creation. They have an evolutionary creation model, which makes no sense. No, in creation, God can front load the genome with as much diversity as he wants to. If all...
Notice, as I said there, they use an argument that assumes evolution to argue against creation. Nothing worse than arguing against a position by starting off with a straw man or a misrepresentation.
All variation is caused by mutation. Absolutely, it's impossible. You can't get all the variation we see in modern people if we start...
Right. If we had to explain all diversity, all DNA differences as the result of mutations, then yes, in a young Earth creation timescale, we would be invoking mutation rates far too fast. These mutation rates would clearly mutate us to oblivion. Okay, it would take millions of years if you assume that all DNA differences are the result of mutation.
From Adam and Eve if they're 100% homozygous.
Right.
But what if God front-loaded Adam and Eve with like 20 million variants? Then we can lose half of them and still have as much as we have today.
Right. So at the bottleneck, as Dr. Carter brilliantly points out, you can lose half of those 20 million variants. You're still left with 10 million, which is actually what we see on the planet today, is roughly, when it comes to common variants, about 10 million. So you can lose 10 million. You can lose half, because they always want to point out that the bottleneck is such an issue at the flood. You'd lose all this diversity. You couldn't explain all the diversity today if you had a bottleneck at the flood. You can lose half, and you'd still have more than enough.
And 20 million is not a lot. I carry 3 or 4 million. You carry 3 or 4 million of the common ones, and probably 3 or 4 million rare ones. That's fine. So it's actually trivial to start with Adam and Eve and get the people we have today as far as the number of variants. Especially if you add 6,000 years of mutation, you can have a lot of new variants appearing in that 6,000 years.
Right. They're coming from their perspective of the evolution having to work in a short time period.
Yeah.
Well, so what is some of the best...
Okay, so let's go over... So that was the first clip, just to show you that most evolutionists, most critics will assume evolution to argue against creation. Now, Swamidass, he has come out against... And I'm going to show you a clip from the Jensen versus Venema debate. 4.5 hours. Can't tell you how many times I've watched it. It's an amazing debate. Must watch. Show you a clip from the cross-examination section on that one regarding this issue. So Swamidass has come out against people like Venema and Collins who use that argument, that make an argument that assumes Adam and Eve were created genetically homogeneous. But unfortunately, he doesn't go all the way there, because he doesn't assume the full created heterozygosity model. So let's go over a couple things there. For anybody who's new to the chat, who just got here, we showed at the beginning a post, or I should say a comment from Dr. Swamidass utilizing some arguments that have been dealt with over and over and over again, okay? And I was just kind of going over the fact that, yeah, Swamidass would at least assume a heterozygous Adam and Eve, okay? But the fact is, based on what I have seen and based on what other young earth creationists have seen, Doki Doki, thanks for the super sticker. Thank you so much. You guys are all awesome. I really appreciate the support. So, but based on what we've seen, he assumes a straw man version of it, okay? He definitely doesn't start by assuming God would have created. This is one of the funniest ones, okay? He doesn't assume that God would have created these design variants in what? As I said earlier, useful, beneficial, and helpful linkage groups. You know, what did God front load Adam and Eve and the created kinds with these functional DNA variants, these alternatives, and then forget to place them in areas within the genome where they'd be helpful and useful? Oops. You know, that's not the God I worship. Okay, so if you've read Dr. Carter and Dr. Sanford's work on this, which he stated that he does, they point out that the design diversity model has actually been validated using numerical simulations. The math has been done, and the allele frequencies, the patterns, I should say, the patterns fits beautifully well in a biblical creation model and starting point of front-loaded genetic diversity. Their must-read paper I've got right here, I believe. Right here, Adam and Eve, design diversity and allele frequencies. So, must-read paper for anybody interested in the technicals of it, of course, the math that they say has not been done. Okay, they also point out, Dr. Sanford pointed it out in genetic entropy. And it sounds like a lot of critics have read that one, but you can find it in their technical work as well. They point out that these design variants would have originated at high allelic frequencies. Then when these designed alternatives are scrambled over time, new traits would be revealed. Okay? New beneficial traits. There is a lot of evidence for front-loading in the genomes of living organisms, okay? Forward-thinking mechanisms abound in the genomes of living organisms. Do evolutionists claim evolutionary mechanisms like natural selection, mutations, for example, have a mind? That's what they would have to do in order to explain that which requires what? Forward thinking. And that which requires forward thinking requires a forward thinker, obviously. Don't get me started on these critics' inability oftentimes to comprehend creationist technical work. Okay, when it comes to the recombination rate and the breaking up of these design variants that they say is an issue, they almost always fail to mention gene conversion. And oftentimes they'll go on this huge tirade saying that it's impossible. And my response is 2 words: gene conversion. I guess you forgot about that. Well, as Dr. Carter explains, and I've explained many times, we got someone new in the chat. Tudu, good to see you. Thanks for joining us. So I've explained that essentially, Dr. Carter explains this, and I believe it's his neutral theory, the Out of Africa paper in the Journal of Creation. He says that essentially gene conversion is a process by which a section of DNA can copy itself onto a highly similar section of DNA in close proximity. It is expected that gene conversion acts over short distances, breaking down linkage disequilibrium between closely linked markers where crossing over is less likely. Oh, and definitely check out my last video on recombination rates, because the rebuttals that I've seen in the Peaceful Science page is exactly what I've predicted. They've been incredibly disappointing. It's nothing but hand waving, storyboarding. It's simple, okay? The recombination rate slows down over time. Why? Well, it's because mutations destroy the PRDM9 sites in the genome. Okay? They want to deny this. We see more sites in Africans, and we see Africans have more recombination. We see less sites in non-Africans, and lo and behold, what do we see? Less recombination. They want to force their human evolution story on us by saying that the genetic diversity, what we see in the genetics of Africans versus non-Africans, is because Africans are older, when in fact that's not the case. These evolutionists are so biased, and quite frankly, I'm getting kind of sick of it. So you can't really make this stuff up. Facts. Crossing over is not random. It is affected by genetic factors. And there are variations within these genetic factors that affect the rate of recombination. It's time to just get over it, evolutionists. There's no problem in our model. The problem is in your model. Why are there still so many large linkage blocks? If we've been evolving for millions and millions and millions of years, what we know about crossing over, what we know about gene conversion, for example, from generation to generation, okay? Our genome should be scrambled, a lot more scrambled than they are, even considering the African genetic diversity, okay? Should be a lot more scrambled. No, what we see, there's large blocks, okay, within the genome, because there hasn't been that much scrambling going on. There hasn't been millions of years of recombination, gene conversion. There's only been a few thousand years. That's why we've got so many large linkage blocks. It's simple. It's easy. You know, we don't need to invent stories. We don't need to invent rescue devices. So let's go over. Brother Sal says, you're the protein structure that you talk about, Brother Sal, in the lectures, and man, it's rock solid. I mean, the evolutionists can't even touch it. You know, though, irrefutable as far as I'm concerned. So George Bond says, it's $10 super chat. George, thanks so much, brother. I'm eating dinner tomorrow. Just kidding. So anybody who just got here, Thursday, be here. Interview with Dr. Sarfati, Evidence for Creation. We're gonna be very interactive in the chat, okay? We're gonna have lots of questions: age of the earth, radiometric dating, evidence for a young world. We'll get into some genetics. It's gonna be fun. George Bond says SFT. Well, let's see. I should start putting these up on the screen. So we got Brother Sal here. Doki Doki. Thank you so much for the super stickers. Doki Doki said SFT is a beast, but the SF team are the reason he is a beast. Amen, brother. I'd be nothing without the team. This is a team effort. And God bless all of you guys for that. Doki Doki says the nail in the coffin is Sal. That's right. That is right. Not going to deny that. You know, it's a great time to be a young earth creationist. It's not a great time to be an evolutionist. not a great time at all. So let's pull up Swamidass's comment there again for people who just got here. And since we've been going 31 minutes, we're going to get right into this, where he says, so we've touched on humans are far less genetically diverse than other species. We touched on why that is the case and why that's what we predict. Then he says, due to the flood bottleneck, young earth creationists should predict that humans are more diverse than any group of land species. So let's, I've got some papers and visuals on this one, but and a couple clips. Okay, but before we get into that, I want to point out the fact that even before I looked into, I would say a year and a half, 2 years ago, when I really got into this, starting to do debates, discussions, doing this channel, just by reading the scriptures, okay, and seeing that God created two human beings, Adam and Eve, and then God created populations of animals, ecosystems of animals, fully functioning ecosystems. Evolutionists, critics, even some creationists sometimes get confused that because God said to, you know, for the most part, bring 2 of every kind, 7 of some, of course, onto the ark, they think that's also what God created. No, God created he created animal types, animal kinds in bulk, in populations. It was only Adam and Eve that were created in 2. So right off the bat, I knew what we should expect then in animals is a higher higher levels of genetic diversity versus humans where we should expect lower levels of genetic diversity because God created two. That restricts genetic diversity. Tony Lincoln says it would be a good start if one of those evolutionists read up on creation material before they enter these debates. Yeah, that's the problem. That's the problem, Tony. They don't, they expect it's going to be an easy win. You know, they think, oh, creation, you know, they must think of it like flat earth or something like that, you know? And then they go into the debate and they're like, you know, they're going up against Sal, and Sal's bringing up protein structure, and he's bringing up the strong genetics, what we know about genetic degeneration. And, you know, we're bringing up our testable predictions and so on and so forth, the things we've talked about for hours and hours, and they're thrown off. They're like, what? How do I rebut this? George Bond says they don't do their homework. Well, George, we've done our homework. So, yeah, so let's get into it. So we've talked about a few things there: gene conversion, recombination, design diversity, what we should expect. Okay, let's go back to Swamidass's arguments. Why don't you know what? I'll put it up on screen so people can at least read it before we get to some more visuals. Okay. What he says here just clearly exhibits he has not read the relevant literature. What do creationists say about animal and human diversity? Well, we've covered that. It's clear from scripture that although God created just two human beings, as I said, Adam and Eve, two human beings, but he didn't just create two of every type of animal. He created populations. Right off the bat, this restricts the genetic diversity of Adam and Eve. But when it comes to animals, and I know I'm sounding redundant here, but it's important because they keep using these arguments. When it comes to animal types, we can expect mostly, I want to say mostly, higher levels of genetic diversity, okay? This higher genetic diversity within animal types would apply to the kind. The kind. And in a lot of cases, that would be the family level. Kingdom, phylum, class. Okay, but here's the thing. When it comes to taxonomy, is it always the family level? Not necessarily. That's why we need to make predictions, for example, retrodictions, and see where it fits. You know, is it the genus, the family, and so on and so forth. But when we look at like dogs and felids, we can probably safely say the family level. So here's the thing, though. This is what I want to say, because I've seen evolutionists point to various species within a kind or within a family. Okay. Now, when you think about the classification system from Linnaeus, kingdom, phylum, class, order, family, genus, species, genus, family. Okay, so there are some kind barriers that are maybe within the genus or the family, maybe the order, especially when it comes to plants or insects. You know, then it gets a little bit crazy. So that's where the predictions come in. Now here, I've seen Erica do it. You know, they'll point to various species within that kind or within that family that have experienced bottlenecks. Bottlenecks reduce genetic diversity. And there's other reasons why genetic diversity can be reduced in, say, a species. But it's the greater, you know, they'll use this as like a gotcha moment. You know, haha, see, we got you here. But they're only pointing to a species within a kind, within the greater kind, where the kind itself has great levels of genetic diversity. It appears that God has designed animals with a greater ability to diversify. This is based on observations, empirical observations. And you know what? Let's go to a visual here. Get rid of this, share screen. Dr. Jensen, and I know Swamidass has seen this debate, or maybe he hasn't. Maybe he hasn't, but I know he's commented on it before. Much highly recommended debate. I mean, look at this, 4.5 hours. I've got it uploaded on my channel, 1,400 views. I've watched this intensely. Dr. Jensen dominated. And it was Dr. Cy Gard in a comment section or a live chat one time, a couple times where I've mentioned this. And I believe he used to work for Biologos, no longer anymore. But he said that Venema would admit that he lost this debate. And Cy Gard admitted that Jensen won. Cy Gard said if he were to debate Jensen, he'd have to prepare a lot. You know, Jensen's a warrior here. So let's see. Exact same question that Swamidass is almost hinting. And I don't want to misrepresent Swamidass. Maybe maybe his arguments here, he slipped a little. He didn't explain himself correctly. And therefore he can, you know, come clean on it. But here is this exact topic. Animals. You know, why do they oftentimes have more diversity? So let's play this. Let's play this section.
Okay, another question. One thing as a biologist that I'm often curious about with the young Earth literature is that...
Yeah, Tony Lincoln says, I saw a debate with Dr. Jensen and the other guy hadn't even read the book. So that was, I think, 6 months in the making for Dr. Herman Mays to read the book, and they were supposed to discuss testable predictions, discuss Jensen's active research program on mitochondrial DNA, Y chromosome, and the history of civilization. And right off the bat, Mays brought up a straw man argument saying that, sure, you're arguing that mitochondrial DNA goes back just 6,000 years. You know, there's not a lot of mutations that separate us from the Eve consensus sequence. And his argument was, but, you know, there's nuclear, the nuclear DNA goes back even further, millions of years, you know. And you can't read Replacing Darwin without missing the fact that Jensen explains the vast majority of nuclear DNA, if not all the nuclear DNA is created heterozygosity. That's why we're looking to the uniparentally inherited DNA, because those differences, we can agree with the evolutionists. We can agree those differences are there by mutations. So let's do a head-to-head prediction. So Jensen kept asking, what does my book say? What does my book say? Which was actually a good question. He was using a very, very skilled method of debating to prove to the audience, to prove to anybody listening that Mays was using a straw man argument that there was no excuse for, because you can't read Replacing Darwin without quickly realizing that the nuclear DNA is the result of created heterozygosity. So that was fun. That's a good point, Tony. That's a good point. Godzilla Freak says he saw the debate but didn't actually watch it. Yeah, that's often Godzilla Freak says, and other people in the comments who also didn't read the book, he says, think that Jensen lost by asking for Mays to actually address his, exactly, yeah. And that's the problem, is Jensen's method was very smart. He was showcasing his debate skills by pushing Mays to answer the question that is answered directly in the book he claimed to read. And that's the problem, is most of the audience, especially the evolutionist audience, hadn't read the book. So it might have come across as, like you said, they were thinking that Jensen lost. Now, I would recommend, and Mays hasn't responded, Jensen did a full breakdown of that debate where he addressed everything point by point. Great breakdown. I think it's called Replacing Darwin on the Debate Stage. Check it out. Check it out. It's a really, really good review. I cite it heavily in my Refuting Critics book. There's been no response. They've tapped out. Caleb Kismet, $5 super chat. Thanks so much. I appreciate it. So yeah, let's get back to this. Okay, this is from the cross-examination section where Jensen really dominated. This was great. So here's the part about diversity within animals.
Many baramins, or accepted groups of relatedness, have DNA diversity within them that are actually far greater than human and chimpanzee differences. Mm-hmm. So I'm curious why that is the case, or how that fits in a young Earth creationist model. If we can,
right? So they'll say that we put humans as their own created kind. They're not related to the great apes like the chimpanzees, the gorillas, for example, the bonobos. But as Vanima is pointing out, there are species within the felid kind, or the felid family, that we would say are related. They descend from a common kind ancestor. But yet those species within that kind or within that family are separated by more genetic differences, more DNA differences than humans and chimpanzees. So he's saying, and this has been brought up too by like Gutsick Gibbon and some of the other better critics, I guess, of young Earth creation, more informed, and they'll say, okay, why why not have humans and the great apes related then, if they're separated by fewer differences than some of the cat species? And those cat species, we would say, are related through common ancestry. And this all comes back to what Swamidass was asserting was a challenge: genetic diversity. We predict lower levels of genetic diversity in humans because God created two, Adam and Eve. Higher levels of genetic diversity in animals because God created animals in bulk. Now let's listen to Jensen's response.
See, diversity in related groups that exceeds
Lagging a bit there,
the
but
human
we're
shape
good.
difference, and from a young Earth perspective, that's happening in a very short period of time. Then why, how is it that we can barometrologically, reliably locate humans as not related to other species?
So the question basically is,
so pay close to attention to the answer. It's a very good answer Jensen gives, and I like Dr. Jensen's style in debates, because what he's doing is he's heard the argument from Vanima. Now he's talking to the audience, and he's going to say the argument is this, and he's going to reiterate it. So now he's not rebutting or answering a straw man. And evolutionists need to learn to do this too. I try and do this. I strive to do this, and it's a very respectful way of debating. So here we go.
if you look at nuclear DNA sequences, there's a whole bunch of ways you can compare it. There's different categories of differences. Single nucleotide polymorphisms would be single rung differences, basically, where I have an A and you have a T. Indels, insertions and deletions would be, I've got a row of these 3 rungs and you're just missing one, missing those 3. You've got whole chunks. It might be, I've got this section of 3 million rungs or 500,000 rungs and you're missing it altogether. These are the sorts of things you'll see. We've got 4.3 million or so, 3 to 4 million single rung differences around the globe, and then a variable number of these other types of differences. Actually, the indels are more like, the small ones are 600,000 or so. All that background to say, let's compare species at the single letter differences. It's easier. And we have data for that, and it's data that I remember offhand, so it's even better. So for humans versus chimpanzees, the single letter differences in the regions you can align, again, ignoring that part that doesn't align, you'd have around, well, you saw it, about 26 million or so, 1% difference. Let's say you compare the tiger to a house cat, which we would say are part of the same kind. There are almost 50 million.
Right. So between the tiger and the house cat, which we would say are the same kind, they're separated by roughly, as Jensen says, 50 million differences. And humans and chimps, if we give them it for sake of argument, if we ignore the copy number variations, the huge gaps, the size differences with the chimp genome, the non-alignable regions, I mean, when you consider all these things and the Y chromosome dissimilarity, they're nowhere near the 98% that they want to purport. But for sake of argument, give it to them, because we are a lot more similar to the chimps than any other animal, any other creature. So give it to them, and they're still separated by, as you said, 26 to 30 million. So why are we concluding that house cats and tigers are the same kind and not humans and chimps? So let's continue here.
Single letter differences. As a general rule of thumb, we'd say that species within a family have a common ancestor. So the great apes belong to a single family. Evolutionists would put humans in the same family. We wouldn't. But chimpanzee, bonobo, the 2 orangutan species, 2 gorilla species, all part of the same kind, we'd say, came from a common ancestor on board the Ark. The orangutan, gorilla, orangutan chimpanzee differences, I think, anyway, they are 3.4% different. I think it comes out to around 100 million differences.
Now, are they all of the same kind? It depends. We can look at the genetics. As I say, genetics is the best way to determine ancestry. I know there's some creation scientists, some leaders in the field that would say that chimpanzees and bonobos share relationship, and gorillas are their own kind. And and orangutans are their own kind. So the question is, are they all related? It kind of looks like, based on the data, especially the Y chromosome dissimilarity. There's a lot of craziness going on in the Y chromosome within the grade 8 family. So let's continue.
So 10 times, well, maybe 5 times higher than what you see in humans. So what does that imply about ancestry? And so
Doki Doki makes me laugh. Doki Doki Bible Club says, Dr. Dennis says, how can I strawman this? Yeah, I don't think he was expecting this. I think that's what's going through their heads, Doki Doki. You're right, Build us a straw man. I like it.
This goes back to the question then of what are the tools we're using to determine it? And are there patterns? Are there numbers of DNA differences? And none of those criteria seem to be good tests. Ultimately, the test is what testable prediction does a model make. So right now, I would say it would seem that God created, let me back up. The data I didn't show you, some of which is in my article series and my technical papers, is young Earth creationists like myself. I've argued that this idea of pre-existing diversity, God creating Adam and Eve with differences within themselves, applies universally among species. That the cat ancestor on board the ark was front-loaded with a whole bunch of different DNA differences, and those then have led to the origin of species. Actually, the primary cause, we'd say, the origin of species.
Remember, if those DNA differences are there from the start, for the most part, we can have mutations that add a little bit of diversity, but all you really need is recombination, reshuffle up those DNA variants to reveal new traits from generation to generation. Gene conversion, for example, can help with that. So these types of processes that evolutionists would agree with, they'll agree that those processes lead to variation, but they have to explain the buildup of the DNA differences and the diversity over time as mutations, which take millions and millions of years. So when they accuse us, old earthers will often do this. They'll say that, you guys believe in rapid evolution. You guys believe in more evolution than the evolutionists believe in, which is so far from the truth, because the evolutionists have to explain the origin of all DNA differences, the origin of all genetic diversity as a result of genetic mistakes over time. Even saying it, is ridiculous. Genetic mistakes over time, to them, can explain all the complexity we see today, all the genetic diversity. No, as young Earth creationists, we'll say that, and it makes accurate retrodictions, predictions. It makes sense theologically, scientifically, that those DNA differences were front-loaded into Adam and Eve. Adam and Eve would have had pre-existing differences within themselves and across. This would also apply to the biblical created kinds. The DNA differences are there. All it's going to take is one generation after the creation event to produce new variety.
So from that perspective, I would say it would appear that God created animals with more potential for diversity than humans. So as it relates to the question of baronology, for many years, the criterion has simply been breeding. And then now, because we're getting DNA data, we can finally bring genetics into it. And so we're now having to develop criterion. What sort of tests can we use to identify common ancestry? Well, what testable predictions does a model make? So if I can say, let's use mitochondrial DNA. Let me think of actually a better example. Bovids. So Bovidae family, 135 species alive today. Everything from cattle to sheep to goats to African antelope. Very diverse family. Cows and sheep differ by 10% of their DNA, I think. So 10 times higher than human chimpanzees. We'd say maybe they have a common ancestor. They're clean animals, so it might get a little sticky, but that's a side note. Mitochondrial DNA, that's where I was going with this. So there's maybe 2,000 differences between cattle and sheep at the mitochondrial DNA level. Humans versus chimpanzee, there's only 1,400. Humans versus humans, you saw the data, 80 to 40. So how do you explain the fact that you have cattle and sheep potentially sharing a common ancestor, but there are thousands of differences apart? Humans, we'd say, have a common ancestor and not related to anything else, less than 100. That'll probably be a function of DNA differences, mutation rates. So no one's measured the mutation rate in these bovid species. We can make testable predictions. And really that's what it's going to come down to. So I would say we've made retrodictions for humans. We can make testable predictions for Khoisan peoples, other people groups in which it is yet to be measured. I can make predictions then for sheep and cattle. And the success of this model will be seen on how well those predictions bear out in reality.
And remember, what's funny is people like Jensen, he's saying, you point at a species in the wild and I'll make a prediction. I'll tell you how fast they mutate, how fast they change. I'll make predictions on speciation rates. But all the rebuttals we've seen so far to the creation model is just kind of nitpicking, complaints, for example. But they're not actually stepping up in making predictions and retrodictions themselves. So animals, it appears that animals were created with a greater ability to diversify. They've been created with more DNA variants. They were created in populations. They were created in bulk. But we still have the potential rebuttal from Swamidass. So let's leave no stone unturned. He says, due to the flood bottleneck, okay, due to the flood bottleneck, young Earth creationists should predict that humans are more diverse than any group of species. And he says Carter doesn't note this. Carter doesn't talk about this. Okay, so let's keep going. I'm going to share screen here on, actually, you know what? Let's kind of break that down first. Okay. Let's also break down kind of what we've heard thus far. Doki Doki, thanks for the super sticker. Okay. First off, as we said, evolutionists were shocked. They were surprised. They never predicted the low diversity humans have. They invented the post hoc ad hoc out of Africa bottleneck. Okay, they require this bottleneck in order to explain human diversity, when in fact our model, which I find fascinating, demanded this. This had to be true. Humans are 99.999% similar. Okay, there's very little variation. And guess what? We have the flood bottleneck. And Swamidass, based on his other question there, must have forgot this one. We have the Babel event. Remember the Babel event? What happens after the flood? The animals migrate and fill the earth. They fill the earth after the flood. They don't congregate in Babel as well and create their own little one world society, and God confuses their languages. No, they diversified right away. They migrated. They filled the earth. While humans, on the other hand, they didn't listen to God. And what did they do? They congregated at Babel hundreds of years after the flood. This would have what? This would have limited the diversity even more. Limited the diversity once again, as this would in a way be a bottleneck, a couple thousand at Babel for however many generations. Therefore, today, we would still predict humans have low levels of genetic diversity, while animal types, and this is exactly what we see, for the most part, there's always an exception to the rule. Okay, the evolutionists want to point to some exceptions to the rule as a rebuttal. That's always their argument. That's always their rebuttal, is an exception to the rule. Okay, when exceptions to the rules are your best arguments, you know you've lost. I'm sorry. So animal types would have more levels of genetic diversity. Look at the chimpanzees. Look how much diversity they have. Look at the felids, for example, as Dr. Jensen and Vanima were talking about there. But we also do have evidence for the flood bottleneck in our genetics. Look at that latest study regarding barcodes, the CO1 gene, that highly conserved mitochondrial gene, where there's low levels of diversity, where they had to once again invoke a selective sweep, invoke some type of bottleneck to explain the low diversity in the CO1 gene. Highly conserved. Those are good genes to look at. I've mentioned that numerous times in debates, including my most recent one with Dr. Garrett. So let's break it down. What are we looking at? Well, it appears that based on all known data, God front-loaded animal kinds with a greater ability to diversify, which probably has a lot to do with them being created in bulk. Now, this means less of their diversity would have been lost at the flood as compared to humans, okay? When it comes to humans, as you've seen Dr. Carter explain, if Adam was front-loaded with, say, 20 million variants and lost half, we'd still have 10 million, which can explain everything we see. There's about 10 million common variants we see on the planet today. We see that in the world today. So let's go to a couple more visuals. An article here. Actually, I took this from, I'm going to screen share, guys. I took this from the book. Boom. See if I can read it from the screen. Make sure you can see this, guys. Evolution's Achilles' Heels, where Carter, this is a great book. This is a really good book. I highly recommend it. So look on screen here. Carter addresses this. Let's read along. If you can follow, that's awesome. So give me the A-OK in the chat when you can. Let me know that you can see this. So let's see what he says. Carter says, interestingly, and as an aside, animals are different. Humans are exceptionally uniform when compared to most animal species. Chimpanzees are 5 to 6 times more diverse. Side note, chimpanzees are more diverse, okay, than humans and Neanderthals. And humans and Neanderthals interbred. By the biological definition of species, they're the same species. They're the same kind. But yet evolutionists want to say they're a part of a sister group, or, you know, they're not descended from Homo sapiens, you know, they're not the same species. They'll say they're not the same species when in fact all chimpanzees are the same species, and they have more diversity within themselves than humans and Neanderthals even do. Are Neanderthals that different? Thank you, Doki Doki. Doki Doki, thumbs up and says cool. Awesome. Thank you so much. So I'm glad you can see it. Let's continue reading. The common house mouse carries huge variations in their population, including multiple chromosomal rearrangements. Examples of this in nature abound in the animal world. Some of this is due to degradation of animal genomes over time. Some of this is due to degradation of animal genomes. Oh, here we go. Some of this is also due to the different starting point of animals compared to people. Remember, as I said earlier, different starting point. The Bible does not say that God created 2 of every kind of animal. In fact, one might assume he created a functioning worldwide ecology that included high diversity. Let's read that again, directly from Carter, who Swamidass said didn't address it, but he was referring to the interview. But I assume that Swamidass was up to date on the literature. So Carter says, the Bible does not say that God created 2 of every kind of animal. In fact, one might assume he created a functioning worldwide ecology that included high diversity. Now, mind you, I pointed this out at the beginning that I understood this right off the bat. Once I read the Bible, once I started reading the literature, reading about the low genetic diversity in humans, I knew that based on the fact that humans were created as 2, animals were created as bulk, this is what we'd expect. And this has been confirmed, corroborated. So there seems to be common agreement here between leaders in the field. So in fact, one might assume he created a functioning worldwide ecology that included high diversity, as also evidenced from the fossil record, right? How much diversity do you see in the fossil record? The diversity is fascinating. That's why evolutionists can oftentimes line up a sequence of animals just based on highly diverse they are. We know there's oftentimes more diversity within the same species than between species. We know that evolutionists have taken a certain species. They've done this before. I believe it was with Tyrannosaurus, where it's the same species, but different developmental stages. You know, an adolescent, a teenage one, an adult one, because there was even some change, some variation within the same species just from developmental stages. They, based on the fossils, they assign them to different species, even though they're the same species, just in different developmental stages. So bones can be deceiving. This is why genetics are important. Fossils, bones found in the dirt, they are low quality science. Okay. Brother Salvador says, Bible does not claim that God created only 2 of every kind of animal. Exactly, brother. You're exactly right. Now, it does say and this is where it can be kind of confusing, is on the ark, you know, that's where 2 of every kind were brought on board the ark, 7 of some. So it's tough to say how many kinds of birds were there, right? So there could have been up to 90 kinds of birds. It's tough to say, but that's where we can make the predictions on speciation rates. Roughly 10 to 12,000 bird species today. That means we'd predict roughly anywhere between 2 and 3 new bird species a year for 4,500 years. And based on we know about rapid adaptive change, just in the last couple of years, we've seen new species of finch on Galapagos Islands. You know, so 2 to 3 roughly new bird species a year for 4,500 years is easy. The predictions have been made. The question for the evolutionists would be, you believe that birds evolved from theropod-like dinosaurs millions and millions and millions of years ago. What is it, 100 million years ago, maybe more? Why are there so few bird species? I debated a zoologist, Adam Heap, a couple times. He'd be the best to debate on this topic, and he had to invoke extinction rates. I mean, you got to invoke a lot of extinction events to explain why there's so few bird species. We're seeing new bird species evolve before our very own eyes due to pre-programmed adaptive change. And the fact is, we see so few bird species stay consistent with our model from the ark. But if birds really did evolve from theropod-like dinosaurs millions and millions and millions of years ago, we should be looking at 500,000 bird species. So, I mean, the numbers are on our side. Okay, so let's see. Where was I here? I love rabbit trails, don't I? Okay, here we go. He created, we read that, but that is important. Okay. So the fossil record. Yeah, the fossil record, you see a lot of diversity. It is true that only 2 of the most kinds of, so here we go. This is what we were just talking about. It is true that only 2 of most kinds of animals were on Noah's Ark, but they were selected from a potentially much larger genetic pool than were people, right? Right? If animals were built, they were front-loaded with more genetic variants, they were built in bulk, functioning ecosystems. Well, guess what? Though, even if there were animals, 2 of most kinds of animals, they would have been selected. So God would have sent, say, the most heterozygous pair. They would have been sent and selected from a potentially much larger genetic pool than were people. Since most of the genomic data available today is human, for obvious reasons, we are far from being able to, of course, we're limited always by the data, the available genetic data. This is a fact. But so far what we have is consistent. So he finishes off by saying, we are far from being able to build a model of genetic history for most animal species. That's why we're now in the infancy, where we got predictions on speciation rates, predictions on mutation rates. So fascinating time to be a young Earth creationist. And then he goes in here, besides the number of common variations found in the human genome, there are untold numbers of rare variations as well. These tend to occur only in isolated populations. Remember the evolutionists, they always say, oh, there's too many variants. And oftentimes they're referring to the immune system, which is designed to change, to adapt. So that makes sense. But they won't point out that it occurs in isolated populations and are simply mutations occurring since Adam and since Babel. He says these include blue eyes in the European population, sickle cell anemia in the African population, and many super rare variations that only occur in a single tribe, family, or individual. Remember, most mutations, okay, they're not going to go to fixation. They're going to drift out. Most mutations are lost over time. This is just what happens. He says this is an indication that we came from a small population in the recent past. We don't share uniformly, in brackets, population level variations, is an indication that our genomes are in rapid decline with a rapid rate of mutation occurring in all people groups worldwide. So the question is, why, especially when they use that, there's too much variations, these hyper variable regions. The question is, it's on our side. And the challenge, this is what I love, is the best arguments that they use can be turned around on them. And that's what's kind of funny about this. So that's Dan's video we'll get to later. But Here, listen to this. Carter points this out. Why isn't there more variation? There's still so little variation, so little diversity in humans, even given the mutations that have occurred post-Babel, post-flood. So let's listen to this clip. This is a great one.
Regions of the genome where they find more than 400. Can this allow me?
Oops, Doki Doki just said no sound. See, we almost had the perfect stream. Let me stop sharing. So what I did was, this is where we got to be careful. I did not click share audio as well. That's always something you want to make sure you do. So Doki, thanks for pointing that out. We'll rewind that a bit. So share screen, boom, share audio. I apologize. And make sure, this is an important clip. This is an important clip. So make sure you let me know if there's any more issues. And I will, let's go back to it. We'll rewind it a bit and then give me the A-OK. Thanks, Doki, for letting me know that.
Were they interbred with us? In fact, they're our ancestors. They're human. They're people. They're children of Adam.
Something similar happened to the mammoth, right?
Okay.
They're very inbred.
So we're going to get right into it here, guys. Give me the A-OK on
the end
sound
point of the mammoths. Yes, on Wrangel Island, and the where they held on the longest.
I see.
Before that, there's actually pretty.
Exactly, Doki. Doki says, let's wait for computer evolution. Yep. Mistakes over time take a single-celled-like ancestor into a whale. Incremental changes, descent with modification, modifying pre-existing structures, that which is already there. It'll take your single-celled-like ancestor into a whale. Just give it enough time. Give it enough time.
Diverse genetics of mammoths. You had the southern mammoths. You had the New England.
Godzilla Freak says, there's so much good content in this one Carter video that even evolutionists' future arguments are refuted by it. It's so true. It's so true. They're using arguments that are refuted directly in the video. They don't even know it.
Mammoths, and then you had the Alaskan mammoths and the plains mammoths. They're actually genetically different populations. But as they were dying off, the inbreeding really, in fact, they shrunk. There was a bunch of small mammoths. Those are the last ones.
Right. What about the hypervariable regions in the genome where they find more than 400 alleles? Can this all be genetic diversity explained by the biblical time frame?
Yeah. In fact, usually when they're talking about that, they're talking about the HLA alleles, which are part of the immune system. Our immune system is designed to rearrange DNA to make new antibodies. Now, those aren't necessarily inherited, but it's a highly adaptable portion of the genome. And look, we've had 6,000 years. There's 7 billion people in this world. If you take everyone's family tree and go back in time, we have all these independent branches in the family tree where lots of mutation can happen. So take a gene, if a gene's, let's say, pick a number, 10,000 letters long. There's 10,000 places for a mutation.
I want to point out, too, remember, guys, this was Damage Control Dan's knockout argument. And we addressed it sufficiently. But here's the thing. The evolutionists, they want to throw a knockdown argument at you. But they're so biased and oftentimes dishonest. I'm sorry, it might sound rude, but this is what I've experienced. Because when you give them a plausible answer and then you not only give them an answer, but you give them an objection and a question in return, they just wave away your answer, and then they don't address your question or answer your objection. So that's the point, is there's been time to account for the accumulation of this diversity in these positions, where especially as he talks about the immune system that's designed to change, designed to adapt. The question is, why is there so little diversity if evolution's true? So let's keep going.
To happen. If one letter change in one place makes a new allele, another letter change in another place makes another allele. It's not a big deal. Of course there's a little diversity. And usually when they say there's all this diversity, they don't tell you what the frequency of these alleles is. Most of these things are incredibly infrequent. They're extremely rare. They're a brand new mutation that happened in Tajikistan or in Patagonia or in Sweden. And it's only one family or in one tribe or in one country or one region. So most of these things are vanishingly rare, to the point where you're like, okay, whatever, it's a new mutation. Why is that a big deal when there's 7 billion people? In fact, it's kind of shocking there's so little diversity amongst us. In fact, we have probably 1/5 to 1/10 the genetic diversity of chimpanzees.
That's a good point.
A lot fewer chimpanzees in the world.
Well, now that you mentioned that, because we know humans have incredibly low genetic diversity, like you said, and you're right, they don't like to point out the fact that the allele frequencies and how rare they are. When it comes to them attempting to explain the low genetic diversity, Dr. Carter, it looks like, right, so remember the diversity that we see in humans, the diversity that we see in animals, it's all exactly what we would expect and predict based on the Genesis account of origins. It's them, and you're going to see Dr. Carter explain this. They are the ones that had to post hoc, ad hoc, explain this away. It wasn't predicted by their model. I challenge them. Show me where you predicted this. Their population bottleneck would have been so disastrous. How many generations, how many thousands of years were these populations inbreeding? Deleterious mutations coming to fixation, leading to rapid and accelerated genetic degeneration. This is another example where evolutionists, they have pointed to the cheetahs as a rebuttal, when in fact the cheetahs are no help to them. Cheetahs are down to 7,000. They're highly degenerate. Their sperm's degenerate. Conservationists are worried they're going to go extinct. Yet this same thing happened to humans, and modern humans evolved out of this and were able to build the space shuttle, build skyscrapers, design some amazing things out of a population bottleneck that was disastrous to the point where it should have led us to extinction. Yeah, this is where we should pull up our picture, the evolution storyboard. I think they look to that out of Africa near extinction event.
Yeah.
Is that out of Africa near extinction event, is that plausible? And what are some of the major problems associated with that, since that seems to be what they look to explain the diversity? Yeah, exactly. Faithful, Honest, and True says 2 of every sort, 2 of every kind. Yeah, so that's the thing. You don't want to confuse the arc, where we're looking at 2 of every kind, 7 of some, for example, with the creation event, where we're looking at fully functioning ecologies, fully functioning systems with populations of animal types, creatures.
Well, first of all, it's a post hoc answer. No one predicted the out of Africa event before they found the data, and they're like, oh, what do we do now? Oh, well, what if our population is reduced to an effective population size of about 10,000 individuals? Now, could have been reduced to 1,000 or 100 or 10? Yeah, but they don't allow that in their models. They only go down to about 10,000-ish. And what if that...
Yeah, Lena Powell, that's a paper I always show. I showed that in my Dr. Garrett presentation, where the majority of variants were picked up in the last roughly 5 to 10,000 years. Isn't that funny? how all of the evidence points right back to what we read about in Genesis when it comes to origins, but then the evolutionists need to rescue device their way out of it, storyboard their way out of it. That's why when you read their conclusions, oftentimes, that's where it's no longer empirical. And the evolutionists will say, oh, that technical paper you're citing, are they a young earth creationist? Well, of course not. They're not questioning the bigger picture. They're looking at the details. They're looking at the empirical data that's not quite consistent with their model. But how do we make it consistent? How do we explain it away? Well, we're not interested in that. We're interested in the actual facts, the actual empirical data. It's just so funny when you read all these papers on like the functionality of these DNA elements, such as retrotransposons, ERVs, or even orphan genes. They build these stories as to how these orphan genes came about, de novo gene synthesis. To me, that's a circular argument. They know these orphan genes exist, taxonomically restricted, highly, highly functional and essential to particular organisms. Okay, no evidence of the ancestral sequences. So they say, okay, de novo gene synthesis. Well, why de novo gene synthesis, evolutionists? And they'll say, well, because orphan genes, look, they exist. We see them. Well, why do orphan genes exist, evolutionists? Well, because of de novo gene synthesis. So it's a big circle, right? Big circle. It's not empirical. It's not scientific. Okay, there's no series of mutations that are going to build an essential gene like an orphan gene, but it's what we would expect based on created kinds, the creation event, or these papers where they invoke co-option for endogenous retroviral function, or the ALUs, for example. Brother Sal has some great stuff on ALUs, contributed to the contested bones section on genetics in regards to ALUs, must read. So, that's not what we're interested in. That's not the story is not empirical, but they want to pass it off like it is empirical.
It stayed in that size for a very long time. They never say how long, but a very long time. Well, then, according to mathematics, you're naturally going to have a Y chromosome atom. You're naturally going to have a mitochondrial leave, and most genetic diversity will be lost. Because small populations, the inbreeding, you get a lot of loss of diversity. That's why we have, breeds of dogs. It's because they don't let them breed with another breed. There's only, I don't know, how many German Shepherds in the world? How many St. Bernards in the world? Not many, just a few thousand of each. I don't know. I don't know the numbers, actually. That'd be an interesting question. But that will dictate to you how much diversity you have. The problem with this is this. There are about 10,000 cheetahs in the wild today. And all the population wildlife ecologists are worried that they're going to go extinct because mutations are increasing.
Lena Powell says, how much time does Dr. Swamidass need to account for the variance? Do we know? Well, here's the thing. This is why they're constantly invoking selective sweeps, bottlenecks. The out of Africa bottleneck they've invented to explain the Y chromosomal line, the little variation in the Y chromosome, the mitochondrial DNA line, the low genetic diversity. So whenever they say that occurred, it's hard to get exact numbers from them when it comes to the out of Africa event, because they're proposing it based on the genetics. There's no evidence for it in the geological column in the record, for example, but you'll get different numbers. What this population was reduced to 2,000, some say 1,000, 10,000, maybe 30,000. And then it's hard to even find an exact number. They don't know how many years would this have had to been the case. 10,000, 100,000 years. Well, this is why it's an issue, because it's not one generation. It's not a one generation bottleneck like our bottleneck at the flood or at creation. We have a bottleneck according to our model at creation, a bottleneck at the flood. Flood's reduced to 8 people. Well, what happens? Well, immediately after the bottleneck, it's followed by rapid and exponential population growth. It's essentially a one generation bottleneck. But their bottleneck, the population was reduced and it stayed like that for a long time, 10,000, maybe 100,000 years, not one generation or 2 generations. That's why the inbreeding becomes a massive, massive problem. It would have been so disastrous. So I just find it so funny that This is where they say modern man came out. But essentially, that would reduce genetic diversity, and it would, to them, right? It's a add-on to explain the data. Sal says my talk on SFT's channel about junk DNA discussed ALUs in passing. Yeah, we gotta have you on again, Sal, to talk more on that, because yeah, you're right, Sal. That was the after show with you and Dan, and I think we even read that section from Contested Bones on ALUs. It's amazing. It's amazing the functionality we're seeing. Okay, so let's keep going here.
Increasing birth defects are increasing, litter size.
Well, Godzilla Freak, here's what destroys their position, okay? Here's what destroys their position, because you mentioned the Y chromosome. The Y chromosome has incredibly low variation. Every single Y chromosome on the planet has incredibly low variation. Our Y chromosomes are nearly identical. And every single Y chromosome on the planet today can be traced to a single Y chromosomal ancestor in the not so distant past. Now, our so-called cousin, okay, we split from the chimpanzee apparently roughly 6 million years ago. Let's look at the chimpanzee Y chromosome. Well, the Y chromosome for the chimpanzee was published, and it was published as less than 70% dissimilar. In the paper, they were shocked. They literally said, this is what we expected between humans and chickens. How far back do humans and chickens go in terms of common ancestry? And not only that, when you actually consider size differences, overall architecture, it's less than 35%. Okay, so then they have to invoke some pretty wild things going on in the Y chromosome. We know the Y chromosome does mutate fast. There's only about 4,500 years worth of mutations in the Y chromosome. Here's the thing. If you have a rapidly changing Y chromosome, okay, everybody in the audience, listen to this. This is detrimental to them. They can't explain it unless they want to throw a storyboard. You have Y chromosomes. Every human Y chromosome, nearly identical. Chimpanzee versus human Y chromosome, when you consider overall architecture, gene content, it's 35% similar, which is ridiculous. The only way to keep a rapidly changing Y chromosome nearly identical in humans today, the only way would be for the Y chromosome to be young. That's it. If the Y chromosome was old, we should find some highly mutated or highly divergent human Y chromosomes on Earth today. We don't. We don't find it. It's the only way. It's actually a fatal blow. They're done. They have no answer other than storyboards. Luca's an evolutionist in the chat. Good to see you, Luca. I like Luca. I find him to be more of an objective viewer on the side. So thanks for being here. Doki Doki says, good job. Thanks so much. It's too much fun, Doki Doki. Too much fun. So let's continue.
Size is decreasing, reproductive incompatibility, genetic diversity in cheetahs is going down.
Tony Lincoln says duck-billed platypus must be the common ancestor between humans and chickens. Yeah, imagine you found the duck-billed platypus in the fossil record, that the evolutionists date back to hundreds of millions of years or something like that. You know, they'd come up with all sorts of stories, interpretations. Fossils are fickle. Fossils are fragmentary. You pick up a fossil and you're an evolutionist, you can sing any song you want. You can make up any story. It's low quality, low confidence science is what the fossil record is.
They're too inbred. If they can't survive about 10,000 individuals, how did we? And if that inbreeding event is a near extinction event, we evolved from the primitive Homo erectus to Homo sapiens in a situation that should have driven us to extinction. You phrase it like that, all of a sudden you realize, like, wait a second, this doesn't make any sense.
I love the way Dr. Carter says that. It doesn't make any sense. It's hilarious. He talks about this in the movie Dismantled that just came out too. And it's like, you've got primitive erectus, who we say are human. We got no problem. They had the capabilities of purposeful navigation. They were in every way, shape, and form human. But evolutionists say Erectus was primitive human, not quite human. Erectus didn't have quite the intelligence as modern humans. So Erectus goes into this disastrous bottleneck. Okay, primitive little Erectus, according to the evolutionists, and out comes modern man. The modern man that has built airplanes, has done wonders. I'm sitting here streaming on a laptop. It's too funny.
And we went from, half monkey, half man to flying to the moon.
We went from a half monkey, half man to flying to the moon. All because of a disastrous population bottleneck that should have driven us to extinction. This bottleneck should have driven us to extinction, but it drove us to design an airplane, to design ways to sequence genes, and to even talk about these things today. No, there's nothing that fits in this story. I challenge Swamidass to explain away this data. Give us some actual arguments, some data, not storyboards. This is your story, this out of Africa story. The diversity, the questions you asked, easily explained, actually predicted by our model. Now explain your story. This is what they don't want to do. Remember, they want to give the objection. We answer the objection, and we give them challenge after challenge regarding their position, but then they don't want to answer it. It seems unfair to me.
Right. In a very short amount of time, going through an event that should have bred us into extinction.
Yeah.
So
The false
it all depends on which syllable you accent in the story.
Well, I agree with you 100% that it's a post hoc, ad hoc rescue device, because they never predicted it like you said, Dr. Carter. And I've heard you point out the fact that this is all exactly what... Okay, so I want to go to this paper right here. Article. Okay. Make sure you guys can hear me. Yeah, I think you can. Look at this. I've been aware of this one for a while. Remember, the critics are not up to date. Let's read this, okay? Remember Swamidass's objections, his questions. Now let's read this. The Bible records a severe bottleneck 4,500 years ago in mammals, birds, reptiles. The flood bottleneck, of course. Followed by rapid expansions. Genesis 9:1, God's command to multiply. After the flood, numerous organisms would undoubtedly have struggled to survive or totally become extinct. This would have been followed by yet another bottleneck specific to who? Homo sapiens at Babel, Genesis 11. As languages were formed and more expansion in humans would have occurred. Read this here. Organisms would have been created with maximum genetic variability. Only 6,000 years ago. Look at this. And would thus still contain much more of this than assumed in a long age model. Here we go. Remember Swamidass. You can't make this stuff up. It's too funny. According to this framework, the least... No, Swamidass says we should predict humans have the most for some reason. No, according to this framework, the least genetic diversity should be found within humans. Due to what? The Babel event, which clearly some of the critics forgot about. You got to read all of Genesis, guys. So let's read that again. It's important. According to this framework, the least genetic diversity should be found within humans due to the Babel event, with greater diversity in what? Greater diversity in mammals, birds, reptiles. The greatest diversity in fish, amphibians, which were not destroyed as completely as land mammals by the flood. Swamidass not coming back from this one. I'm sorry. But here's the thing. I love these guys because they give me job security. I'm just kidding. But seriously, their arguments, it's like just one comment that they make, it can lead to a 3-hour refutation video. Additionally, organisms with shorter generation times would be expected to be more diverse as heritable mutations occur between generations. This is a good article right here. Dr. Jin, he's got a whole series. Dr. Jinson pointed out the fact that those from BioLogos and Critics, Swamidass is not affiliated with BioLogos right now, but he says they don't read the literature. You know, you can even see this in the Jinson-Venema debate. Look at this. Okay. Conversely, the nuclear DNA starting point I just described, this is Jensen, easily accounts for the millions of DNA differences that exist among humans today. Copying errors are unable to explain the vast, this is true. And here's the thing, those that want to scoff at the front-loaded diversity model, guess what? Take the abiogenesis challenge then. Refute any of our videos with Dr. Stadler, Salvador Cordova, Dr. Ryan Hayes. We're going to have Dr. Sarfati on Thursday destroying abiogenesis too. Give us a more plausible scenario for origins then. Because we know abiogenesis is dead in the water. No one's taking the challenge. Professor Dave attempted, professor quote unquote, and Dr. Stadler came on and educated him. Professor Dave will never be able to come back from that. So those that want to not argue against our actual position and just say, oh, there's no evidence for this model of created heterozygosity, even though the observations are in accord with it, it leads to testable predictions on mutation rates, speciation rates, DNA function. They're the ones that have no explanation for origins. They've got chicken and egg problem after chicken and egg problem. Okay? They've got no explanation. It's dead. Take the abiogenesis challenge. So look at this. Copying errors are unable to explain the vast amount of DNA differences we see today. How true? How true? Because the vast majority of these DNA differences are not the product of error, but deliberate design. Exactly. Exactly. How do you build these DNA elements that are like, you know, I'm going to go here. This is a good place to go here. Look at this. Hopefully I can read this or I'll just pull it up in my thing. Multiple overlapping codes. Follow along here. I'm going to pull it up in the book so I don't strain my eyes too much, since I got to do this almost every night because we've got ridiculous comments coming from the evolutionists. Listen to this. How does evolution build these types of things? Okay. This is Achilles' heels, page 64, Carter. There is a considerable amount of data compression in the genomes of all higher organisms. Any given section of each of these genomes might be doing several things simultaneously. A single letter of DNA might be part of an exon that is in turn used in 20 different proteins. This is science fiction, I'm telling you, what's going on a genomic level. At the same time, he says, that letter might be part of the splicing code that tells the cell when to produce each of those proteins. Evolutionists, how do these evolve? We don't want storyboards. We want empirical data. That letter also might be part of the histone code that the cell uses to know where to wrap the DNA around certain protective proteins called histones. That letter also might affect the 3D structure of the DNA. It might be part of the pervasive epigenetic code, right? How do you build that, which requires forward thinking, evolutionists? And it might be part of a 3-letter codon that is translated into a specific amino acid. Because the genome is base 4 and because of codon degeneracy, the designer was able to select from alternate codons when faced with fulfilling multiple simultaneous requirements. Degeneracy allowed him to overlap multiple genomic commands without having to compromise the design requirements of the proteins. Multiple overlapping DNA and RNA codes defy naturalistic explanation. Hey, naturalists, materialists, evolutionists, explain this within your worldview. Once again, we don't want storyboards. And make it impossible for natural selection to operate as an agent of long-term evolutionary change. Selection sees the short term, guys. How do we survive till tomorrow? It doesn't see the long term. It can't build these types of mechanisms. Selection runs into a wall of insurmountable difficulty when faced with mutations that affect more than one trait simultaneously. Here we go. This is what I was talking about. I want to get to this. Polyfunctionality means that a given mutation can affect completely unrelated traits, say color vision, the ability to tolerate garlic, and mitochondrial efficiency. Although this would be an extreme example, this is yet another Achilles heel of evolutionary genetics. How could a simple process of trial and error, always seeking the simplest answer to an environmental problem, create an interleaved and multi-layered system of regulation? In fact, this system is one of the wonders of the universe. Isn't that right? One single cell in your body, more advanced than the most advanced and well-designed automobile, more advanced than the space shuttle. Without this level of multitasking, the genome would have to be much larger, and it might not be possible for DNA-based multicellular organisms to exist at all without it. How do you explain this, evolutionists, naturalists? It's, like I said, it's science fiction what's going on in the genome. Data compression into every single nucleotide. It's fascinating. Here's the thing. This polyfunctionality means that one mutation, it's polyconstrained, the genome. And one mutation can affect so many different things, as you read here. So Brother Sal says, that's right, Professor, quote unquote, Dave. If Dave is a professor, then I'm the department chair of dean of the school. Yeah, exactly. Doki Doki says, Sal, Dave is the lab assistant's assistant. I challenged Dave a few times to debates. He won't do it. He's retired, apparently, from Creationists, getting exposed. Oh. Laughter is the best medicine. That's why I love having evolutionists around. They give me a lot of laughter. Okay, so let's see here. Similarly, it appears that God created, this is back to Jensen. Okay, it appears that God created nuclear DNA differences in the animal kinds as well. Genetic calculations suggest that God created tens of millions of DNA differences from the start. Here we go. Look, in other words, the cat ancestor was likely created with one DNA copy different from the other DNA copy, with perhaps 50 million DNA differences. That means more diversity would be retained. You could lose half. Okay, humans at the flood could have lost half, and they'd still be left with 10 million variants if they were front-loaded with 20. Okay? Therefore, however much was lost in the animals, they're still going to retain more from the original front-loaded diversity, Swamidass, okay? The Babel event as well, you're forgetting. Animals migrated. They diversified first. They didn't congregate in some one-world society, one-world government like humans did, and God had to confuse their languages. No, they diversified. They migrated. Humans, because of Babel, would restrict the diversity even more. This, to be honest with you, it's just the model. It's what the origins account of Genesis would purport, predict, and retrodict. So since only about 1,660 years passed between the creation event and the flood, most of this variety, okay, have a look here. Watch this. Since only about 1,660 years passed between the creation event and the flood, most of this variety would have been carried on board the ark. And being so shortly removed from the perfect creation, it's not hard to believe that significant population growth in these animal kinds occurred. He talks about massive amounts of population growth can happen in a very short amount of time, even in slowly reproducing species. Here's the thing. Let's read this. Under this scenario, straightforward population modeling, okay, here we go, indicates that very little of the created genetic differences would have been lost prior to the flood. They would have retained a lot of that initial created heterozygosity, okay? Once the animals stepped off the ark, their reservoir of DNA differences could have easily translated into a massive amount of morphological change. This fact is best illustrated by a consideration of a recent objection to the young Earth creation view of speciation. Over a decade ago, one of the main complaints that could be levied was based on the function of the DNA letters. Actually, it was the apparent lack of function of DNA differences. When the sequence of the billions of human DNA letters were first elucidated in 2001, many in the scientific community concluded that the vast majority was junk, useless leftovers from the human evolutionary process. Presumably, similar amounts of junk existed in other species as well. However, this claim was not based on rigorous laboratory testing of the billions of DNA letters in the human DNA code. Instead, it was based on appearance. To evolutionary scientists, the DNA looked as if it were non-functional. Remember, if evolution is true, for one, they didn't predict as many conserved regions as we see today. If we've been evolving, okay, descent with modification for millions and millions of years, based on mutation accumulation, based on change, we should be seeing evolution expected to see these regions mutated, a lot of randomness going on, and then they've seen that there's so much conservation going on amongst the animal kingdom. And conservation, when you got conserved regions, that means function. You know, natural selection's preserving it because it's important. Okay, they didn't predict that. They didn't predict what we now know on a genomic level when it comes to activity, when it comes to function. If evolution is true, there should be a lot of evolutionary leftovers, a lot of junk, a lot of vestigial DNA, a lot of broken genes, a lot of parasitic DNA. And this is what they assumed. And this is, evolutionists don't want to make predictions on DNA function. You ask them, and they don't want to give it to you because they don't want egg on their face again. They've already been shown to be wrong so many times on this. Since 2001, a large body of laboratory evidence has accumulated. While preliminary and largely biochemical, this evidence has a trajectory suggesting that the vast majority of human DNA is in fact functional. Now, of course, just like Dr. Carter said the other day, just as we believe here that based on genetic entropy, okay, genetic degeneration, of course, we would find a lot of broken genes. You know, the entire genome does not have to be functional because there has been genome degradation. So let's see. With a few more years of experiments, the evidence for function will likely increase. And you can continue reading this. You know, this is a good series. But point is, the literature, I mean, it's here. It's there. You know, they're not up to date on it. So let me see. Go through here, see if there's another clip. So we did the Venema clip. We did. Okay. So as you can see here, that, and this isn't the first time we've addressed this data. Okay. This isn't the first time that we've rebutted these critics. You know, they're running on arguments. The arguments are not really looking too good. You know, I'll screen share here too, because even let me see, in my screen share, boom, see what page it is. Hope everybody's doing good in the chat. Diversity. So I addressed this one about okay, let's share screen, make sure it works. So in my book here, okay, so here's my book, The First Couple, Adam and Eve. Let me see, diversity, pages 29 to 30, so I can find it quick. 29 to 30, I address this too. Actually, I have a whole chapter on this pretty much. I know these guys' arguments, and I like to be ahead of the game. Okay, let's see right here. As we know from scripture, so I'm writing this here, Refuting the Critics. As we know from scripture, there are a few events that would be considered bottlenecks. Let me check the chat here, make sure picture's good, everything's good. Good to see everybody here. Sal says my talk at SFT on junk DNA. Yeah, go check. I think I made a playlist, must-watch interviews. I think it's must-watch interviews / lectures. And I've got a bunch of must-watch ones in there, obviously given the title. And you can find Sal's talk on junk DNA. I think a couple of his talks in there. You can find Dr. Carter's, Dr. Stadler's, Dr. Jackson, Dr. Ryan Hayes. You can find all the good ones in there. Bill Morgan, we had a great interview with Brother Bill. Time flies by, Bill. I seen you in the chat, but that was probably a year ago we had that. We got you in there. Dr. Dino got an interview with him in there. Yeah, so go check that out. Sarfati is going to be here Thursday, so that's where you can find the good interviews, lectures. So, as I say, this would be the creation event, the flood, and the Babel. Okay, the bottlenecks. I have personally engaged in well over 50 live debates, and an incredibly common objection to the biblical model of ancestry has to do with these bottlenecks. They have made the argument that the flood bottleneck, remember, would have reduced levels of heterozygosity to a point that speciation and adaptation post-flood and post-Babel would have been implausible. Well, we've already seen that no, it would be very plausible. It actually fits the data. So I continue. This is an argument that I've dealt with in my previous books. If you have read these books or seen my debates, you would know that the bottleneck at the flood was only one generation. Remember, as we talked about earlier, and it was followed up by rapid and exponential population growth. Extraordinarily little of the original created heterozygosity would have been lost based on these facts. I cite Carter here. Dr. Robert Carter has also addressed this argument in the same article that was previously cited. Source: does genetics point to a single primal couple or response to claims to the contrary by biologos? But yet they're still using these arguments. Funny, eh? Another part of their error comes from the fact that the authors substitute an evolutionary bottleneck for the biblical one. In evolutionary mathematics and theory, one rarely considers a single generation bottleneck. Rather, a bottleneck is defined as a restriction in the population size that lasts long enough to have an effect. A single generation event, for example, Noah's flood, with a rapid expansion of the population afterward, prior to and after Babel, would lead to a negligible loss of diversity. Here's the thing. These guys, you know, sure, someone like Swamidass doesn't agree with the young earth creation, but at least, I guess, understand the model if you're going to debate it, or and that's the thing, you know, you're not going to find people like Dr. Carter, you know, strong men of God, warriors in the faith, even wasting time against critics who misrepresent, who are not bringing forth honest objections, really, you know, because after a while, answering the same questions over and over again becomes ad nauseum. So let's see. Dr. Carter states the three founding ark couples could have carried a huge percentage of the pre-flood genetic variation, Adam and Eve's diversity plus pre-flood mutations, with the exception of the Y chromosome, total loss of all but one line, and mtDNA restricted to 3 lines. Hence, the biblical model fits the available data nicely, and the data do not serve as easy refutation of Adam and Eve. So there you go. So this is the PDF, the first couple, Adam and Eve, independent origins, refuting the critics, biblical creation. So we address all of that in there too. I think we've tackled that one pretty good. Let's see. Let's go back to Swamidass. So here's his thing, guys. Have we addressed his arguments? Let's see. Tell me what you think. Swamidass says, humans are far less genetically diverse than other humans. Due to the bottleneck, though, humans should be more diverse, while groups of land animals and species should be less. And then he asks, why is it that there is less diversity in humans than, for example, and he goes over a number of kinds. And he says, it's interesting that Carter doesn't address this. So as you guys, I think, can see in the chat, we've answered all of that sufficiently. 2 hours worth of answering that. So what we should say then is next. What is next? What will they offer next? People who just got here, I see we got 23 people in the chat. We've been going for a couple hours now. Interview with Dr. Jonathan Sarfati, Evidence for Creation. Be here Thursday. So that one's going to be great. This one's going to be a lot of fun. Okay, so let's end it here. This one's going to be quick. This was the bulk of it. I just wanted to, I'm sick of this whole argument that, there's a lot of activity in the genome. That's what we see. But, it's mostly spurious, or it's not useful activity. Let's deal with that. I'm going to share screen. Let's listen real quick to one of our favorites. So that was 2 hours. I wanted to go into detail on that one because that's an incredibly important one. So I challenged Swamidass to address it. Okay, so you're going to see here, ENCODE takeaway. High percent of the genome exhibits biochemical activity. But then Dan, our favorite critic, he's going to...
ENCODE is that a high percentage of the human genome exhibits biochemical activity, like 80-ish%. But very little of the genome has a well-documented function. It's something we've tested and we know exactly what it does. And that's around 10% of the genome, give or take. We also know that very little of the genome is conserved. So for example, approximately 5% of the genome is conserved across all mammals. That conservation is very strongly indicative of function, but obviously most of the genome with biochemical activity is outside of the conserved region. We know that much of the activity is low frequency, and that is indicative that it is probably spurious rather than having some selected function. So the conclusion is that much of the genome is non-functional, and this is based on ENCODE's data and what ENCODE says about their own data. So to summarize.
Maybe this was wrong because I could do another 2 hours on this one. I mean, so much wrong there. I wonder if people like Dan would put on paper, I wonder if they would put themselves on the line that the vast majority of the genome is non-functional. You know, we're making very specific predictions on functionality. Now he's saying it there, but would he actually put it on paper and predict it? Do a head-to-head prediction, you know, based on future knockout tests, based on future genomic function tests, experiments. You know, do you actually predict that the vast majority is going to be non-functional? So you've seen it there, right? Most of it spurious. Yeah, a lot of activity, but it's still non-functional. Yeah, conserved regions. Yes, he didn't mention why. He didn't mention that it wasn't predicted by the evolutionists that there is as much conserved regions as there are. Let me know, guys, if you could hear, if you heard that though in the chat, the audio, because I know it was a little bit low there. I should have turned it up. But they never consider this. Okay, and I talk about this in my book too. I've debated this topic over and over again. They never think about the fact that, think about embryological development, okay? Picture the complex sequence of events that actually changes a single-celled egg into a complex organism with hundreds of different cell types in different organs, all of them in the right places. Well, guess what? The programming for this has to be somewhere in the genome, and it appears to be in, well, quote unquote, junk, the junk DNA regions. But junk DNA paradigm is dead. Okay, regions that are not conserved, there's, oh, don't even get me started on the functionality. The functionality, and a lot of times it's species specific. You know, we're going to get into that. But there's actually been reports of mice, okay, where the researchers, they actually found that a certain class of, you know, quote unquote junk DNA comprising of like 1/3 of the mouse genome is involved in embryo development. We know so very little about what really goes on down to a molecular. Let's go down to a molecular level. Okay, a molecular level when it pertains to the developmental stages, embryological development. There are so many developmental windows, guys. There are so many stages in development where genes are turned on, turned off. There's so many things going on. It's actually unimaginable. Like we read earlier with the multiple overlapping codes, it's science fiction what's going on in our genome on a molecular level. Okay, so it's unimaginable to know for certain what every single DNA sequence is doing during these various windows, these various stages. What is the DNA doing? Okay, evolutionists, tell me, what is the DNA doing step by step, stage by stage during embryological development? A lot of the function is taking place during these stages, during these windows. And yet Dan here wants to say that the majority of the genome is non-functional. There is so much interconnectedness going on, guys. We have many RNA genes. I pointed this out in my debates with Erica. There's so many RNA genes, even pseudogenes actually, that comes to mind, that are expressed only under various certain circumstances. How many experiments would need to be performed in order to determine for certain if a gene sequence has absolutely no function? Will they go on the line? Will they put it in print? Will they make the prediction that the majority of the genome is non-functional? And here's the thing. I point this out too, for sake of argument. It's so funny. Even if I pointed this out in my second debate with Erica, I think I went right over her head because we argued for 30 minutes on genome function. She was obviously wrong. She was proven to be wrong. And I said, even if, Erica, the genome was 10% functional, like you're saying, that's still too much function. Still too much function for ape to man evolution, because that means out of those 100 new mutations per person per generation that are accumulating, 10 of those are deleterious and detrimental. It's still too much. But yet we know it's only pseudoscience to say that the majority of the genome is junk. Brother George Bond, thanks so much for the super chat, says one person's junk is another person's treasure. Yeah, amen. Brother Sal says, great comments tonight. You guys are awesome. Chat is on fire. Sal says, repetitive elements have biophysical properties that are important for forming the 4D nucleome. Amen. Amen. He says, Dr. Dan didn't touch that issue in my second debate with him. Nope. He wouldn't answer the question as to whether or not man is degenerating either. Is man getting better? Answer that question. Great points. Sal says 4D nucleome affected by ERVs. Man, just think about how much time we've spent on all the various functions. They're going to say that all this function, okay, everything about the developmental stages, as I was talking about, okay, is all just crammed within 10% of the genome. Oh, these evolutionists make me laugh. Just like Speed of Sound, we played that clip over and over again. You know, it would be beneficial if man were to get dumber and dumber. We got to have Sal on again to do a junk DNA presentation one of these days. So here's the thing. Okay, even look at these pseudogenes. Actually, before we even get into this, you know what? I'm going to play a clip here, if I have it, of Dr. Carter. Let's see, guys. Bear with me. Take a little intermission here. Boom, we're going on 2 hours. And I do want to get through this one too. But this one could be another couple hours. I want to go to Dr. Carter. I wasn't planning on this one, but he does make some good points here on. So bear with me, guys. Have some fun in the chat. Good to see you. We got a lot of people here. Bear with me. I want to go to. We talked about junk DNA in this one too.
But we would have predicted from a biblical creation.
So let's find it. Bear with me. Make sure you can hear the audio.
A lot of them have blue eyes.
Salvador says L1 and B1 repetitive junk DNA important for 4D nucleome. Yeah, you know, this stuff's amazing. And to claim, like, I couldn't believe his point on like conserved regions, you know, the non-conserved regions. I mean, these regions are highly functional, and the activity, yeah, you know, we're going to get into that. This bulk of the argument is that a lot of it's spurious. Okay, it's not useful activity, and we're gonna break that down. I want to just demolish that. But before I did, I just really wanted to touch on the fact that it's not a sophisticated way of thinking. Okay, when these guys claim the majority of the genome is non-functional, or although there's evidence for genome-wide biochemical activity, It's not useful. It's spurious, they say. It's just there, but it's not doing anything. We're going to demolish that. But I want to, let's keep this interactive. Let me find this clip, guys. Chat it up in the chat. I'll find where it is. Let's see.
And that's all
Bear
that is.
with
Allele
me.
frequency is just, you look at the frequency of the different letters in the genome.
So we're going to find.
You can go.
He makes some good points on it.
Even though it's not a creation.com product.
I think it's coming up here. He makes a good point on the
what I wrote called "How to Think, Not What to Think," and I put a diagram in there of. But as they did more and more work, they realized that all that was garbage. It just doesn't work. Now there are some groups that have a high rate of one versus the other because of inbreeding, because they have a small founding population. That's fine.
Okay, I think it's coming up here, guys.
If it's just.
Thanks for sticking it out today. I think we gave it to Swamidass pretty good for a couple hours there.
Either have or don't have.
There's a lot to get through, and we did it
on the outside of your cell. I don't know even that's a broken gene. I think it's just a variant.
There we go. I think it's the next one here. Give me the A-OK on audio too, guys. The $10 super chat to ask it. Thanks, Jamie.
It's a wonderful model for medicine because it's a mammal and it's easy to breed. And 5 different places in the brain, different places in the liver, the skin, the tongue, all different organs. And they looked at which RNAs were being expressed.
There we go.
It's a wonderful model for medicine.
Okay, let's listen here, guys.
Because it's a mammal and it's easy to breed.
Is it safe to say
no one really cares
the junk DNA paradigm has been overturned?
They're not bunny rabbits. Oh, little bunny rabbits, and they're monkeys, right? they're just mice, and they're using an amazing amount of scientific experimenting. So, anytime we have a lab model from a different species that we've developed, it doesn't mean it works in humans. Doesn't mean it would work if you find a pesticide that kills these mice. It doesn't mean it's going to kill the mice out in the wild. But it gives you an indication. Oh, this is something that does this. Now let's test the next step and see how far it goes. A lot of times that kind of science doesn't go anywhere, but most of our cancer drugs, for instance, have come out of things like that.
I see.
So,
what have you discovered with the telomere length in human beings? Have you noticed that you said Europeans are most inbred? So would you find that most telomere length is shorter in people like us?
A massive new project just stopped. It's a 10-year program called the GTEX Consortium. When they first sequenced the human genome, we're like, we'll understand disease. Yeah, that wasn't true because the genome's a lot. They took tissue samples from like 800 something cadavers, multiple places in the body, like 5 different places in the brain, different places in the liver, the skin, the tongue, all different organs. And they looked at which RNA. They took tissue samples from like 800 something cadavers. Multiple places in the body, like 5 different places in the brain, different places in the liver, the skin, the tongue, all different organs. And they looked at which RNAs were being expressed. Well, that's cool. That's which part of the genome is active. And then they said, okay, we have all these things being expressed. What letters does this person have versus that person? And oh, that letter controls this gene. Oh, and all of a sudden we're understanding function in the genome that we haven't ever understood before. When they first sequenced the human genome, we're like, we'll understand disease. Yeah, that.
Right. This kind of function was not predicted. And oftentimes you'll debate these evolutionists and they'll say, well, we never predicted all the non-coding regions were junk. Yeah, they might have predicted 1 or 2 here and there, functional RNAs. But the amount of function that has been discovered was never predicted.
That wasn't true because the genome is a lot more complicated than anyone thought. But now we're finally figuring out, and one of the papers, it was in Science 2 weeks ago, they looked at telomere length, and they're able to see that men have different telomere length than women, that Africans have different than Europeans, that telomere length changes with time, with age, and different tissues have different telomere lengths, but the only one where it really changes over time is in the testes, because telomerase enzyme is active in the testes and not other places. But if the length of a telomere, it basically controls how many times the cell can divide. Because every time a cell divides, the telomeres get shorter. if you have short telomeres, you're probably not going to live as long as someone with long telomeres. I don't know if you guys can hear that, but we just came up with a giant Georgia thunderstorm. It's pouring rain and thundering just outside my window. So if power goes out, bye. Anyway.
Well, no problem, Dr. Carter. Actually, you know what? Before I ask this one question, I'm curious because you mentioned ENCODE and the hat map data. I got it. Okay, here we go. We found it, guys. We found it. Intermission's over. Although there are some good points there too on function. So here we go. ENCODE, DNA function. Then we're really going to take on this whole spurious claim. And a good comment in the chat. Sal says, I prefer to say it's a bad working assumption to say it's junk. That's right. I agree. It's an example of how evolutionism is bad for medical science, utterly useless. Yeah, can't agree more. Can't agree more. 100% useless. Medical science is important. And to assume junk that you're looking, if you assume junk, how hard are you going to be looking for function then? How hard are you going to be looking for cures to these diseases? To say I love your lecture on that one. Watch that a few times just to understand it as best as I can. But when it comes to like ENCODE and let's say junk DNA non-coding versus coding, is it safe to say for the most part that the junk DNA paradigm has been overturned based on what we now know?
Absolutely, but the evolutionists won't admit it.
Right.
So is it safe to say? Sure. Is it safe to say in public? No, you'll be ridiculed and called stupid and you don't understand what's going on. But because they're not going to let go of it. See, it doesn't matter. There's so many areas where this is important. One of the important parts is the difference between humans and chimpanzees.
Right.
See, if we're 99% identical, they can explain that in evolutionary time. If we're 96% identical, they're in trouble. If we're 95%, they can't do it mathematically. If we're 90%, forget it. We're not evolutionary related to chimpanzees. Or gorillas existed with T-Rexes.
Right.
And they're not going to do that. You can't, stretch out because, once you learn all these differences, you have to say chimpanzees and humans are further back in time, our common ancestor. But there's only so far you can go. So they haven't been willing.
Right. Here's a huge point that Sal says. Huge point. He says 90% of single nucleotide polymorphisms associated with disease are in the what? The non-coding regions. Great point.
And it pushed back beyond 6 million years. Or else apes are with dinosaurs, which they don't like that idea. Anyway, the ENCODE project, can you hear that?
Not at all.
No.
Okay. Wow, that's amazing. All they did was they looked at 1% of the genome, scattered in little bits and pieces all over the genome. And they found out that almost all of the parts they looked at were made into RNA. Now, that means it's functional because you're not going to have 99% of your genome making parasitic RNA. You're not going to waste all your time making all this junk because that'll just gum up the system.
Could you imagine, as he's saying, gumming up the system by just wasting all of this energy? We're talking about DNA. We're talking about the cell here. Wasting all this energy just transcribing useless, worthless, non-functional evolutionary leftovers, parasitic DNA. What a waste. Not very efficient. Not very efficient at all. But this is because of deep time evolutionary assumptions. We don't assume that. Those in the medical field working towards medical advancement, we've heard it over and over again. We've heard it from Brother Sal, people that are directly involved in this, Dr. Stadler. They don't assume junk. It's a bad starting point. It's a bad assumption, junk, non-functional DNA. Doesn't make any sense.
Biology is very streamlined. It's not inefficient. So if we're making all that, we must have a function. And that was their argument. And now the GTEX consortium is saying that, oh no, it's a lot worse than ENCODE said. There's a lot more functionality to the genome and a lot of RNA being manufactured that actually does things. It's not random.
A lot of RNA being manufactured that actually does things, is essential, is functional. It's not just random. But according to Dan here, Dr. Dan, most of this activity is spurious. It's not really functional. We can only be certain that the conserved regions are that which is functional. Now, I don't want to put words in his mouth, but I just found that argument was weird. you know, the whole conserved versus that which is not conserved. There wasn't much of an argument there.
I don't remember what the question was. I was just rambling.
No, that was a perfect answer. And it makes me think about how many. I've got a good, let's see, since we're screen sharing, I notice I put another one here. Okay, I'm going to flip to this page too, so you can see it on the slide. Let me expand it for you guys. Maybe this will help. Let's see, stretch it out. Ooh, no, that didn't help. It's made it look worse. Okay, well, there it is. I'm going to flip 57. Let's read this. I'm going to flip it so it's easier on the eyes. Okay, let's check this out. Make sure you can hear me. Wow, look, you guys are party animals. Antonio, brother, good to see you. Lena, good to see you're still here. George Bond. You guys are night owls like me. 3:45 in the morning. We're having fun. We could do this all night. Let's read this. Also, functions have been found for what much of the vast stretches of non-protein coding DNA that sits between genes. It turns out that most of the genome is active. The ENCODE project was a multi-university, multi-million-dollar, multi-year study designed to determine how much of the human genome was transcribed, turned into RNA, a measure of function. They only analyzed 1% of the genome. That's what he was talking about here. But they included both protein coding and junk DNA regions in the analysis. This project demonstrated that on average, any given letter of the genome is used in 6 different RNA transcripts. This does not mean everything is turned into protein. Far from it. It also does not mean that everything has a mandatory function or even that the letters are used often. What it means is that most every letter does something. This is important, okay? Since form follows function is a general rule in biology. Form follows function, right? The fact that these regions are active. Now, contrary to what Dan says here, it's not spurious, it's not useless activity, okay? The fact that these regions are active strongly suggests they have a function. And we've made predictions. I want to see them putting in print predictions. I want them to actually put their cells on the line and predict the majority of the genome's non-functional. Okay, let's continue. Why else would the cell allow so much transcription, right? Isn't this key? The cell dedicates a significant portion of cellular resources toward making non-protein coding RNA. Thus, the cell would be greatly benefited by turning off. Let's read that again. We need to teach Dan here. We have become the teachers. Thus, the cell would be greatly benefited by turning off this waste of energy. Natural selection over millions of years would have killed off the parasitic RNA processing. Now, of course, they're going to pull out the storyboard, force fit it into the story, as they always do when they don't predict things, and they fail over and over and over again. Natural selection, I'll read it again, over millions of years would have killed off the parasitic RNA processing. It has not done so because it is necessary. See, isn't this is the more parsimonious explanation. It is not done so because it is necessary for cellular function. In fact, the genome can now be seen as an RNA computer. Now, they can't admit this, that the non-coding regions function much like the processor in computer code. They can't admit this. They'd be too close for comfort to a designer. They don't want to fall on their knees and worship the creator who've designed these amazing systems. You know, we worship the creator. Praise the Lord. The general lack of junk DNA is yet another Achilles heel of evolutionary genetics. For without it, evolutionary mathematics does not work. And more functions are still being found for the non-protein coding DNA. In fact, the junk looks like it is more active than the genes. Oh, this is what's funny. This is what really destroys Dan. The junk looks like it is more active than the genes, turning the old idea that we are protein-based organisms on its head. In the words of an evolutionary biologist, J.S. Maddock, says, the failure to recognize the full implications of this, particularly The possibility that the intervening non-coding sequences may be transmitting parallel information in the form of RNA molecules may well go down as one of the biggest mistakes in the history of molecular biology. Let's read that again. May well go down as one of the biggest mistakes in the history of molecular biology. It's like a play. You go watch a play, you're watching the actors and actresses on stage, right? Doing their thing, performing, doing their job, fulfilling their roles. You know, that's not all that's going on. What's going on behind the scenes, right? Like the non-coding regions of our genomes. There's so much going on behind the scenes. You've got people that are coordinating. They're helping the frontliners, you know, on stage, the actors, the actresses, those that are working the lighting, those that are working the technicals of the stage, those that have written the script, whatever, you name it. There's a lot of people backstage doing the thing. You know, this is interconnectedness, coordination. Evolutionists often seem to think that it, you know, if it's not producing a functional protein, it's not functional or it's worthless. Oh, I mean, there's so much more going on in the genome than was ever imagined. So this whole argument from Dan, let's, you know, I got to play it again. It's just, I think, you know, this kind of stuff needs to be exposed. Let's go back. What did he say here? Boom. Let's see.
Functionality in the genome. In other words, there's a lot of junk DNA in the human genome, according to.
I just realized maybe I'm not sharing the audio, so I better go do it again. Share screen, share audio. Okay, let's see. Boom. Make sure you guys can hear. Now listen up. After everything we've just read and talked about,
a percentage of the human genome exhibits biochemical activity, like 80-ish%. But very little of the genome has a well-documented function. It's something we've tested and we know exactly what it does. And that's around 10% of the genome, give or take. We also know that very little of the genome is conserved. So for example, approximately 5% of the genome is conserved across all mammals. That conservation is very strongly indicative of function, but obviously most of the genome with biochemical activity is outside of the conserved region. We know that much of the activity is low frequency, and that is indicative that it is probably spurious.
Low frequency, probably spurious, high percentage of biochemical activity, just not useful, not much going on. Here's the thing too, even with the let's say pseudogenes, a lot of this so-called junk DNA, okay, it just being there, the persistence of them, let's say the persistence of these so-called non-functional regions, okay, is in itself further evidence of their activity, okay? And this is a fundamental challenge for evolution. It's a problem. It's a hurdle. This is why they got to resort to storyboards in light of the empirical data, as well as the logical conclusions, okay? As we know, and what we know about natural selection, as it would be expected, natural selection would remove these types of DNA sequences and DNA elements. This also goes for so-called, let's say, junk ALUs, junk retrotransposons, the so-called parasitic DNA. If it were actually useless, or the activity is really just spurious, and yet what we know about DNA, that since DNA itself is manufactured by the cell, transcribing all this useless junk is energetically costly. Because of the lack of selective pressure on this, you know, neutral DNA, one would also expect that these, say, pseudogenes, according to the evolutionists, that are older, okay, they should be scrambled beyond recognition as a result of accumulated random mutations. It's not real empirical science to say that the majority of the genome is non-functional, or that a high percentage of this biochemical activity is just spurious, or it's just, low frequency, not really useful activity. Let me share screen again here. I address this too in great detail, but one specific section that jumps out. Let me see. In my book here on page, see if I can find it here, 95. So let's go. So 95, this is the Refuting Critics book. Another argument employed by those that want to hold on to the junk DNA concept, right? Because they want to hold on to it. They know they need it. Creationists and evolutionists have opposite predictions when it comes to this. Okay, so I say the junk DNA concept is that this activity detected in the genome is not useful activity, as he was saying. Why would the cell bother with so much useless junk? I've never actually heard a good answer to this. I have had answers, more storyboards, or an answer that's based on the assumption of junk DNA, based on the assumption of deep time evolution. This would be a waste of time and energy for the cell. These so-called junk sequences, including the retrotransposons, would be mutating to oblivion. The evidence in every way is against junk DNA. As I iterated earlier, evolution hinders scientific advancement. Evolutionists must maintain that the greater portions of the genome are made up of evolutionary or genomic fossils and evolutionary leftovers. The philosophy of evolution can no longer be reasonably defended. I've said it numerous times during the course of this book, and I'll say it again. Human evolution has been overturned. Let's see here. In the article titled "More Nails in the Coffin for Junk DNA," Dominic Statham covers many more fascinating functional roles found in the so-called junk DNA. Here we go. Remember this? Critical to the correct growth of embryo to baby and the subsequent correct working of the body is the timing of protein production. The right genes must be switched on at the right time so as to produce the right amount of the right proteins when they are needed. All of this, all of this just crammed into what, 10, 15, 20%, according to these guys. It is being discovered that the DNA controlling the use of genes lies in areas formerly deemed junk. It is now known that one gene often produces many different proteins, and the junk DNA interspersed within the gene contains information needing to facilitate this. Moreover, junk DNA can modify the way chromosomes, right, packages of DNA are organized, thus changing the way the DNA functions. Some genes formerly deemed to be functionless and therefore labeled pseudogenes have now been found to function and actually produce proteins. Fancy that, eh? Other pseudogenes are now known to switch off genes when they are not needed, or to increase their use when more proteins are required. So look at this right here. There was an article I was reading. This one is an older one, but there are still some interesting points to be made here. So let's see. Okay. I mean, every day, just more and more on this. Okay, so DNA that appears to lack conservation of sequence is probably nevertheless functional in terms of general isochore composition, serving as background for proper gene function, as a spacer between genes and their long-distance regulatory elements. And see, his argument here on the conserved regions, which I need to emphasize they didn't predict so much. It should have been all just a scrambled mess if we've been evolving for millions and millions of years. Okay, but the conserved regions, like he said, are indication of function. You know, we have a lot of shared sequences due to shared function across the entire animal kingdom. Okay, similar morphology, similar anatomy, physiology, genetics demands similarity in genomic sequences for functional purposes. This is good design. This is actually necessary design. So he says these areas that appear to lack conservation of sequence act as spacers between genes, and they're long distance regulatory elements. And in the case of introns, in terms of physical length alone, monotonous DNA repeats probably serve a regulatory function for certain genes. Let's see, this paper after paper you can read, but you can just find little articles You know, you don't have to do, this one's news-medical.net. In genetics, the term junk DNA refers to regions of DNA that are non-coding. Some of this non-coding DNA is used to produce non-coding RNA components such as transfer RNA, regulatory RNA, and ribosomal RNA. However, other regions are not transcribed into proteins, nor are they used to produce RNA molecules, or their function is unknown. The importance of these non-coding regions has been a key focus in genetic research for many years, particularly in terms of evolution. Of course, they... So here, comparative genomic studies have shown that some of this junk DNA is highly conserved, sometimes over hundreds of millions of years, which suggests that these non-coding regions have undergone positive selection throughout evolution and confer an advantage in terms of preserving or minimizing certain biological traits. Let's see here. For example, the human genome and the mice genome. So this, once again, assuming evolution, but here. However, the protein coding DNA only makes up about 20% of the conserved DNA in these genomes, with the remainder of the conserved DNA found in non-coding regions. Researchers have also found that some sequences in the DNA that is junk act as genetic switches, which determine where and when genes are expressed. There's millions of these genetic switches just waiting to be turned on and off via environment, via triggers. And as we talked about before, many of these non-coding RNAs are only expressed under certain conditions, like the pseudogenes, for example. It's just science fiction what's going on a molecular level. Some regions of the non-coding DNA may also be essential for chromosome structure, the function of centromeres, and play a role in cell division, meiosis. Some non-coding DNA sequences also determine the location where transcription factors can attach and control transcription of the genetic code from DNA to mRNA. So here's the thing. It's pseudoscience to say that the majority of the genome is non-functional. The activity, they'll say, as you heard there, is just spurious, not useful activity. Why is the cell bothering with it? It's a waste. It's not effective. It's not an effective use of resources. Plus, we're the ones making exact predictions, specific predictions. We're in the infancy of understanding the DNA language. Okay, we've got specific predictions on mitochondrial DNA proteins. Okay, the highly conserved ones. We have DNA function related predictions when it comes to hierarchies. Okay, and based on the fact that we understand so little of the DNA language, so little of the developmental process, embryological development, it's premature for any evolutionist to assume that the majority of the genome is junk. Assume treasure, and you'll find treasure. Anyways, let's have a look at the chat. We've been going 2.5 hours. I think I managed to touch everything I needed to. So still got 20 people in the chat. Lena Powell, Luca, good to see you here. Yeah, so good to see everybody. Thanks for Brother Sal. Thanks for sticking it out. 2.5 hours. We got through. I had a lot of things I wanted to cover. I wanted it interactive. Yeah, just like Sal says, exactly. Yeah. I think there's probably some junk DNA because of genetic entropy. I've seen some credible evidence of some real pseudogenes. Definitely. Yeah. We wouldn't predict that the vast majority, we wouldn't predict that it's all functional based on what we know about genomic degradation. We're living in a fallen world, and this is what would be expected, but no way we would be predicted that the vast majority is non-functional junk, reflective of evolutionary lefters. Nope. Just enough that would reflect genetic degeneration. Moving up in the chat here, make sure I seen. Yeah, Jason says, yeah, 6,000 new diseases added into the database every 3 months. And they get 1 or 2 mutations that are somewhat positive, but mostly degenerative in these 1 or 2 so-called beneficial mutations. In a few people groups, it's going to select out all the other 5,998 diseases that are being added to the world population every 3 months. This is one rescue device for genetic entropy. George Bond says, non-coding by evolutionary terms is junk DNA, or maybe you guys are coming around. So, yeah, they never predicted nearly close to what we find. But yeah, so good to see everybody. We've been gone for a couple hours. We challenge our critics to represent the position correctly. And guys, be here for Thursday. Dr. Sarfati is going to be here. We'll be interactive in the chat. So you guys got questions? Come prepared. Thursday, 3 p.m. EST. And as you can tell, SFT versus Swamidass. He's been corrected. The evidence, guys, it's a great time to be a young earth creationist. So I'm pretty tired, gonna get some sleep. Good to see everybody. God bless, and probably see you tomorrow for something, but definitely Thursday. Looking forward to it. God bless. SFT is out.
For all those who appreciate the work that we're doing here on Standing for Truth, please